Hello everyone, this is Doug, and in this lesson we're going to introduce you to carbohydrates and monosaccharides. Now, when most people think carbohydrates, right, they think of those delicious food group. So to be the base of your diet, right, if you compare them to fats and proteins. And so most people think of like these plants, right? Like I have a picture of wheat down here. As a chemist, I think of these molecules, like I have cellulose drawn up here as a bunch of rings with oxygens and hydroxyl groups and stuff all over it. But when people talk about sources of carbohydrates, right, the big one is plants, right? When you say starches, people are talking about things like amylose and amylopectin. We'll talk more about amylose in a couple videos here. And also like fibers. If you look on a nutritional label and they say fiber, you know how many grams of fiber there are in a serving. They're talking about a bunch of molecules, but fibers, one of them being cellulose, for example, which is this molecule right over here. That's a snippet, I should say, of cellulose. Which is supposed to be mostly indigestible. We'll talk more about that later. And then you've, of course, heard the term sugars, right? So where sugars are supposed to be the small things that are really rapidly digested by you. You may have heard terms like complex carbohydrates and simple carbohydrates. These would be the complex ones, the large molecules, complex carbohydrates. And they're called complex because they're bigger, and also the idea is that they are metabolized more slowly. I'll put metabolized slower. As compared to, right, if you had sugars, right, so like glucose, sucrose, which would be simple carbohydrates. So you got simple carbohydrates, simple carbs. The idea is that those simple carbohydrates are metabolized much more quickly. So I'll put metabolized faster, which has a lot of health implications. You guys probably all have heard about diabetes and American diets being too high in sugar and carbohydrates and stuff like that. We're not going to get into type 1 and 2 diabetes and insulin and everything like that. We're just going to talk about what carbohydrates are, how we recognize them, what are the main types. And then in animals, we have glycogen. So animals don't create amylose, animals don't create cellulose. We store everything as glycogen. So that's sort of our main way to store carbohydrates. And we'll come back to all of these in a lot more detail in a second. I'm just kind of trying to lay out a little background information for you. Some of the functions, again, when people think carbohydrates, they think of eating it to give us energy, which is absolutely true, right? You eat a potato, it's going to give you energy because you're going to metabolize that starch into glucose, and that glucose is get transported around, put into a cell. You'll undergo cellular respiration, glycolysis, citric acid cycle, the whole shebang. So that's absolutely true, right? There's definitely a lot of energy production. For plants, though, there's also a lot of energy storage, which is why there are starches there in the first place, and in us, right, in our glycogen. Again, we'll talk more about how we store a little bit of carbohydrates in our liver and muscles. But they're also really important. Carbohydrates are also really important for cell-cell signaling, and that's something that I don't think people really talk about that much, but it's super important. We're not going to go into like immunology or anything like that, but the idea is that, you've probably heard of blood types like A, B, and O. That is based on what sugars are being presented on the outside of your red blood cells, outside of your blood cells. So if you have certain sugars pointing out your type A, if you have certain sugars pointing out your type B. And so those A, B, and O differences are just based on the types of carbohydrates that are on your cell membrane. Now that plays a huge role, right? You might have heard that if I have blood type A and you try to give me a blood type B, I'm going to have a huge immune response. And that's because the immune system also relies, not entirely, but in part, on recognizing two different cells as either being from the same organism or as a foreign entity, right? Should it attack your cell or should it not? And it uses what carbohydrates are presented on the outside of your cell. So carbs are also really important for this cell-cell signaling or cell-cell recognition. It's kind of how they talk to one another, right? It's kind of like the braille on the outside of a cell. When they bump up and they kind of feel around, they can tell, is this cell from the same organism, from your host, or is it from something outside of the body? Or in other words, is it some sort of pathogen? Is it something that needs to attack? So carbohydrates are really important, not only for energy, but also for cell-cell signaling and cell-cell communication. Now, if we look at carbohydrates, the original word, right, came from this idea that you had carbon, so carbo for carbon, and then hydrate as in water, because it was observed that a lot of these carbohydrates had a formula where you'd have one carbon for every one water molecule. And so they called them carbohydrates, because things tended to have formulas like C6H12O6. Whoops, let me erase that. C6H12O6. C6H12O6. Right, or in other words, this is like saying that you have 6 carbons and that you have 6 water molecules, right? Hence the H12O6. And that's fine, and that's true, and that was an observation, but that's largely a historical definition. I mentioned this before. Really, for us, what we want to look at when we hear carbohydrates, we want to think polyhydroxy, aldehyde, or ketone. So polyhydroxy, right, meaning that there's many hydroxyls. So we need to find many hydroxyl groups. And the second criteria is that it's an aldehyde or ketone, right? So we need to have either an aldehyde, C double bond O with an H on it, and then whatever. Or we need to have a ketone, which is a carbonyl, right, C double bond O with 2 carbons on either side. It needs to be those 2 functional groups. So I got 2 criteria, the stuff in green, the stuff in purple. Those are my 2 criteria. And it's a much more specific definition of carbohydrate than just generally a ratio of carbon and water. There's lots of things that are C6H12O6 that are sugars, but that are carbohydrates, but there's lots of things that are C6H12O6 that are not carbohydrates at all. I'm not going to draw them all out, but just trust me that just because something has a formula that has a ratio between carbon and water in it does not make it a carbohydrate. So I have these 4 examples down here, and I want to know, are these carbohydrates or not, right? Are each of these 4 things carbohydrates or not? So I'm going to look at my 2 criteria, right? So my first thing, is it polyhydroxy? Well, I see that I have 2 hydroxyl groups, so check, that works, right? So I have 2 hydroxyl groups, nailed it. And I need to know also, is it an aldehyde or ketone? And so if I look at this carbonyl here, it looks like I have a ketone, right? Because I have the C double bond O, and that's a carbon and carbon on either side, so that works also. So that means that, yes, this thing is a carbohydrate. This would be considered a carbohydrate. This is dihydroxyacetone for people who care. It's the smallest ketose. But regardless, I have a ketone, and I have many hydroxyl groups, so I'm good to go. If I look over at the second one, yes, I have multiple hydroxyl groups, right? In this case, I have 3 hydroxyls. I have 3 hydroxyl groups. And if I look at my carbonyl functional group here, it looks like I have a carboxyl, right? Because I have a C double bond O with an OH on it. And that's where my problem lies, right? Yes, I have hydroxyl groups, but I don't have an aldehyde or ketone, right? The carboxyl is not an aldehyde, nor is it a ketone. So that means no, this is not an example of a carbohydrate, because it does not have an aldehyde and it does not have a ketone. It doesn't have either of those. So that would not be considered a carbohydrate. Here I have something that has, yes, it has 4 hydroxyl groups. That's great. 4 hydroxyl groups. However, I don't see any, right? I don't see any carbonyls whatsoever. So no, it does not have, so I'll put here no carbonyls at all. No carbonyl group, so no aldehyde, no ketone. So this also would not be a carbohydrate. Now, as a side note, for those who are just kind of curious about things, I am. This is the structure of erythritol. Erythritol, which is one of those alcohol sugars. You may have heard of alcohol sugars like xylitol is probably the most common one. Where alcohol sugars are things that look kind of like sugars, only they only have alcohols in them. They don't have those carbonyls. They don't have ketones or aldehydes in them. So they kind of taste sweet, but then they don't get metabolized like true carbohydrates do. So they taste sweet, which makes us happy, but they don't get turned into calories typically, or at least not many calories. So they make our gut and our body mass happy. And then if we take a look at this last one, yes, I have hydroxyl groups. Looks like I have 3. I have 3 hydroxyl groups, and then if I go here and I take a look at my type of carbonyl, this looks like an aldehyde. So now I have met those 2 criteria, right? This would be considered a carbohydrate because it does have an aldehyde and it does have 3 hydroxyl groups. So that's what I'm looking for, right? If I ask you which of these molecules is a carbohydrate, that's what I'm looking for. How does it have multiple hydroxyl groups? Is it an aldehyde or ketone, right? That's our definition. Now, if we take a look at our three main monosaccharides here, right? So if you look at the word monosaccharide, mono, right, just means one. So if I look at mono, right, just meaning one. And then saccharides just means something sweet. So it's the sugars, it's the sweetness. So we're talking about sugars or a sweet thing. You may have heard the term like someone's being saccharine. They don't mean the artificial sweetener. If someone's being saccharine, that means they're being overly sweet, like to a cheesy degree, like, let me count the ways I love you, and, you know, like, just overly sweet if someone's saccharine. So the word saccharine means like a sweet thing or like sugar. So monosaccharides are talking about things that are just one sugar. Now the three main monosaccharides are these. First we have glucose, which hands down is the most important monosaccharide. It's just everywhere. Basically all organisms make it. It's also known as your blood sugar. Right, when people talk about, your blood sugar has gone up, that's what they're talking about glucose levels. It's also known as dextrose. It's also known as dextrose. And it can be utilized basically by every cell in the body. So it can be used by every cell. Right, every cell has like some sort of GLUT transporter, like GLUT1 or S, or depending on where you are in the body. But glucose can definitely be brought into like all of the cells, and that'll be different than some of these monosaccharides we talk about in a second. Now, if you take a look at glucose, right, hopefully based on our previous discussion in the slide right beforehand, right, if you look at glucose, yes, it's polyhydroxy, right, because I have all of these hydroxyl groups, so it's polyhydroxy. And if I look right here, I have an aldehyde. So it looks like glucose should be considered a carbohydrate, which makes sense, right? That's why I'm talking about it right now. So aldehyde. Now, if you take a look at glucose, we can number these carbons. I'll number these just for reference in a second. But we always number the carbons for the monosaccharides, starting with your carbonyl as carbon number 1, or the end of your chain with the carbonyl as number 1. So then I have number 2, 3, 4, 5, and 6. So it's got 6 carbons. And as you've probably seen before, right, if you counted all of these things, it looks like for glucose, I do have 6 carbons. And if I count the number of hydrogens, I get 12. And if I count the oxygens, I get 6. So I get C6H12O6, which is why people often say that is the formula for glucose. Now, if I take a look at the next monosaccharide, the next really common monosaccharide, this one is fructose. So here we have fructose. And fructose is often found in fruits, right? Found in sweet things like sugarcane, honey. You might think fruits. I'll put honey. I'll put agave. High fructose corn syrup, people think of, so high fructose corn syrup. A big difference between fructose and glucose is that not all cells can use fructose. So fructose, most of the fructose needs to go to the liver to be transformed into either glucose, which is fine, it gets sort of transferred out as glucose goes into the bloodstream, or as fatty acids, or as fats, basically, which is kind of problematic. You don't really want your liver shooting out a bunch of fats. They make VLDLs, which is a very low-density lipoprotein, tend to crash a lot and cause heart disease or contribute to heart disease. And you don't want to get a fatty liver also. So some of those fats don't escape the liver, and you can get a little bit of cirrhosis, kind of analogous to what you see with alcohol consumption, but not nearly as extreme. And so fructose looks like this. It's not an aldehyde, right, like glucose. It looks like this functional group here instead is a ketone. So it has a ketone in it instead of a aldehyde. But it's also, right, if you take a look and you start counting, if you take a look at this, it also has 6 carbons, 12 hydrogens, and 6 oxygens. So fructose also has the formula C6H12O6. So just because you're C6H12O6 does not mean you're glucose, does not mean you're fructose. It just means that you're something that has those, right? It's not super descriptive. But you'll notice, right, since these both have the same formula but they're different compounds, right, remember that these are called isomers. Fructose and glucose are isomers of each other, right, because they have the same formula but different structures. So same formula, different structure. All right, they look pretty different. And then lastly, if we go over here to our last one, that's galactose. So we have galactose. Galactose you're primarily just going to find in things like milk, like mammal milk. And we'll talk more about lactose in the next video. But it's part of lactose. It's part of your milk sugar. And that's easy to remember because it has the word lactose in it. It's galactose. Galactose is another example of an aldehyde. And if you look at galactose, it looks a lot, right, like glucose. It looks super similar to glucose. And if you compare these two structures, there's only one place where there's a difference, and that looks like it's at carbon number 4, right? Carbon number 4, we change the OH from being on the left in the right on glucose to the left in galactose. That's the only difference between those two. So they're epimers. We change just that OH there. But you'll notice, right, since it's so much like glucose, it's also C6H12O6. So that means all three of these are isomers to one another. So this is also an isomer. So again, like I mentioned in a previous video, right, the molecular formulas are not super descriptive. I could draw so many structures with C6H12O6. These are just 3 of them. These are just 3 sugars. There are lots more monosaccharides out there. There's allose and mannose and all kinds of things. But these are the 3 really big ones, the 3 really, really common ones. So these are 3 that I want you to know for sure. Now, I drew all of those monosaccharides on the previous slide in their straight chain form, their linear form. Sometimes you hear it called the open form or the linear form or the straight chain form, whatever you want to call it. I usually call it the straight chain form. But you'll also see all these carbohydrates in rings. And so I want to talk about how you go from the straight chain to the ring here. In this picture, I just drew glucose again, so here's your glucose. I'm going to go ahead and I'm going to number those carbons, again, like I did on that previous slide. So here's my carbon number 1, number 2, 3, 4, 5, and 6. And now what happens with glucose, for example, is that the OH here on carbon number 5, it actually attacks carbon number 1 to make a new bond. So this long chain sort of wraps around to make a ring. It's sort of like taking a long piece of rope and looping it around and tying it so that your rope is now circular. And when you do that, so this one in green, I'll just kind of try to highlight this in green here. That's this O right here in these rings, where the O in these rings are from carbon number 5. And if I transfer the numbering over, what you're going to see always in this type of drawing, which is called the Haworth projection, or Haworth. So the Haworth projection, that's this style of drawing your carbohydrates. Always, 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 you're going to see 1, 2, 3, 4, 5, and 6. Those are those carbon numbers. Oh, it looks like this one's slid out. Oh, that's a bummer. I'll redraw this one. That's a bummer. I should pay more attention when I'm making these slides. So let me redraw this real quick. Right, so then I'll call this carbon number, excuse me, number 1, 2, 3, 4, 5, and 6. And so we're always going to be able to do that. We're going to be able to start with the rightmost carbon and go clockwise, 1, 2, 3, 4, 5, 6, for all of these that we see. So you guys can just kind of ignore this for right now. Now, what we're going to do here, right, what I'm missing from these is I'm missing my OHs. I didn't put the OHs on all of these. And so what I need is at carbons 1, 2, 3, and 4, I need to put these OHs on here, right? If you look over here, carbon 2 has an OH, carbon 3 has an OH, carbon 4 has an OH. And the way you do that is you just take the original straight chain form, how it's drawn like this, and just have it fall down and flop over to its right. So in other words, I'm going to draw glucose something like this, right? Where I'm going to have it fall over so that I have carbon number 2, carbon number 3, carbon number 4, Carbon number 5 was here, and then this last thing was OH and a CH2. Right, so I just tipped it over to the right is the idea. I just had it fall over to its right. Now if I go back and I look at, well, what should be going on at carbon number 4 here, right? If I want to know what's going up and what down, at carbon number 4, it looks like, and let me label these again, oops. If I look at these numbers again, so that's still carbon 1, still carbon 2, still 3, still 4, still 5, and 6. If I look at what's going on at carbon number 4, looks like carbon number 4, the OH is going down. So when I go over here, I'm going to draw my OH going down. So on number 4, I'll have my OH going down and the H going up. If I look at carbon number 3, it looks like the OH is heading up. So when I go over to my drawing and I draw, well, where's the OH? The OH is up and the H is down. If I go to carbon 2, on carbon number 2, the OH is down. And so again, when I go over to my ring form, I'm going to draw the OH as down. And now what's kind of weird here is that carbon number 1, I need to draw an OH either up or down. But the problem is, right, I don't have an OH here. Number 5 attacked number 1, and so there's no OH. The OH is neither up nor down. It's going to be one of those two. There's not like definitely up or definitely down. And so you can draw it either way. I can draw it down, or I can draw it in the other form, which is where it's up. And then here I'm just going to add the rest of those OHs. Right, it's the same molecule, so I'm just going to add the OHs the same. So I can have two different forms here. I can have this top form where the OH is down, or I can have the bottom form where the OH is up. So here the OH is down. Here the OH is up. That's the only difference between these two. And the idea is that these two molecules, right, when this glucose forms, they can form either one of these two. When the OH is down, this is what we call the alpha anomer, the alpha form. So the alpha form or the alpha anomer, where anomer is the term that's used for these types of differences, where the only difference is that OH or up-down when this open form closes. When the OH is up, this is called the beta form or the beta anomer. So these two molecules, they look really, really similar. They're kind of like isomers of each other. They're just specifically called anomers because they close and they open to form this. And this closing and opening process is called mutorotation. So the idea here, right, is that you have glucose in a straight chain form. It might mutorotate to give you the alpha. Here, let me draw this. Right, so you can imagine that glucose might close and it might give you the alpha anomer. And it might be the alpha anomer for a little bit. And then the idea is that alpha anomer could open back up to give you the straight chain form. But then maybe when it closes, now maybe this time it forms the beta anomer. And then the beta anomer opens back up to give the straight chain form. And then it closes again to do the alpha, and back and forth and back and forth and back and forth, which is what this double-headed arrow thing is supposed to mean. And so that process of going back and forth is called mutorotation. And this isn't unique to just glucose. It's also the same thing happens for fructose, galactose. Really any sugar can mutorotate. And so no one can hand you, for example, a bottle of alpha glucose because it's constantly, as soon as you put it in solution, it's going to open and close and open and close and open and close. Now there are versions, one is more stable than the other one. We don't have to get quite into that right now. But we definitely want to know whether or not our OH is going up or down, because it's going to be hugely important for when we start identifying what types of alpha linkages we have. Are they alpha linkages or are they beta linkages? So pay close attention to, is your OH heading down from carbon number 1 or heading up from carbon 1? And so we'll start talking about that in the next video when we talk about disaccharides and glycosidic linkages. So if you have any questions or comments, please leave them down below. Happy studying. Take care.